Welcome to Ask the Expert. It's a pop-up digital cafe for scientists who study type 1 diabetes and interdisciplinary fields. And today we're here with Dario Gerace, PhD. He's located at Harvard. He recently was given the, I think the best abstract at EPITA. Is that correct?
Yeah, that's right.
Yeah, pretty good. He began his training in cell and gene therapy for type 1 diabetes in 2011 at University of Technology in Sydney, Australia, in the lab of Professor Anne Simpson. And his early work was focusing on the in vivo transdifferentiation of pig hepatocytes into insulin-producing beta cells. He then received his PhD in 2017, also at the University of Technology in Sydney, where he was looking at the differentiation of mouse mesenchymal stem cells into beta cells. And then he moved to Boston to Doug Melton's lab at Harvard as a postdoc, where he's currently working on the genetic engineering of immune-evasive human stem cell-derived islet cells. So basically trying to find and create the invisibility cloak for islet cells so that they can be transplanted into those who need them, candidates such as those who have type 1 diabetes.
That's exactly right.
Yeah, so welcome. So happy to have you, and love to see some slides.
Yeah, of course. Thank you for the invitation. Let me try and share my screen here now.
Okay.
See how we go. I hope you can see my screen.
It's we can see it perfectly. Looks
perfect. Great. Okay, well, once again, thank you for the invitation, Monica. I've been following all the Sugar Science Ask the Experts videos on YouTube, and they're always very interesting and very informative. So I'm happy to be here to share some unpublished data. I've been in Doug's lab now for about 4 years, working on this problem of engineering immune-evasive human stem cell-derived islet cells. And I think this is probably one of the hottest topics at the moment in the field of beta cell replacement therapy.
Agreed.
And yeah, and in fact, I think ViaCyte just released, did a press release on some really interesting clinical trials that they're going to be doing with their gene edited lines. So it'll be interesting to see, the safety and efficacy of those trials. But I'll start off now, I think. And I like to start with this slide because it really shows the proof of concept as to why we're interested in generating stem cell derived islet cells. And this slide essentially tracks glucose concentration in diabetics pre and post islet transplantation. And so before islet transplantation, you can see that their blood sugar levels vary quite significantly throughout the day. So this is throughout the time course of a day. And in gray here is the normal glycemic range. So this is really where these people want to be. Normal people are within this range. And in fact, after islet transplantation, you can see that this works quite well in normalizing their blood sugar levels. And so this really is the proof of concept as to why we want to use stem cell derived islet cells as a cell replacement therapy. And the reason being is that islet transplantation is limited by has two major limitations. The first being that it is limited by the number of donors. So islets need to be harvested from cadaveric donors. And in most cases, you need to harvest islets from multiple donors to treat an individual. And the second problem is that they are also then required to be on lifelong immunosuppression, which comes with a whole host of nasty side effects that we kind of want to avoid. And so I think we've probably addressed maybe the issue of shortage of supply by generating stem cell derived islet cells. And this slide here kind of summarizes a good 10 to 15 years of work trying to essentially coax a pluripotent stem cell or an embryonic stem cell through the developmental pathway that leads to the production of beta-like cells. And in fact, at the end of this differentiation protocol, we end up with a heterogeneous population of endocrine cells, which include alpha cells and some delta cells and some off-target cells as well, which I think at the end of the day, we're going to also try to eliminate from these products. But ultimately, the goal is to then transplant these cells into individuals with type 1 diabetes, such that it then cures their diabetes. And I think Vertex also have released recently a single patient that was dosed with half a dose of their therapeutic dose of their stem cell derived islet cells, which showed some really significant improvements in the quality of that person's life.
Yeah.
But I think the problem now that we're trying to solve is the immunogenicity of these cells. So we'd like to put these cells back into a person, ideally without any encapsulation, and to avoid immune rejection of these cells. And so there are two main strategies that many labs, including our own, are pursuing to identify novel gene edits to induce hyperimmunogenicity. And the first strategy is to use CRISPR screening, where we introduce a lentiviral CRISPR knockout library into Cas9 expressing beta cells, transplant these into in vivo into mouse models, and then perform longitudinal in vivo cell imaging using bioluminescence imaging, and then ultimately recovering the grafts that survive. And ideally, these would be the cells that have been protected from immune rejection, and try to understand what edits are unique to those cells that have provided them with that protection. The second strategy is more of a targeted engineering approach. And this is the approach that I've taken, which is to kind of take to adapt immune-evasive mechanisms from many biological phenomena. And these include tumor biology, viral evasion, fetal maternal tolerance, and microchimerism, and try to engineer each of these individual strategies into an ES cell or IPS cell that would then ultimately be differentiated into stem cell-derived islet cells. And on this slide, I've kind of just summarize some of the main and well-known pathways that we can interfere with to induce some type of tolerance or immune evasion. But in fact, we're not the only ones that have been interested in this. There have been a number of papers now that have been published on generating hyperimmunogenic stem cells. And in fact, recently there were two papers, one from Ron Evans' lab and one from Matthias Hebrick's lab, that nicely showed that you could potentially protect these stem cell-derived islet cells by engineering them either to overexpress PD-L1 or by eliminating some class 1 molecules from the surface of these cells. And so, but one of the problems with these strategies, though, is that many of them suffer from transgene persistence issues. So you try to introduce your transgene of interest into your embryonic stem cell, and then after differentiation, in most cases, what you find is it's either not there at the end of your differentiation, or you have mosaic expression of your transgene of interest. And then post-transplantation, those transgenes could potentially be further down-regulated. And so what we want to develop an engineering strategy that will result in persistent expression of our transgenes such that the immune-evasive phenotype that we generate can be persistent in vivo as well. And so essentially, we solved this by targeting the expression of luciferase to the GAPDH locus. So the GAPDH gene is a constitutively expressed gene. And we reasoned that if we targeted our transgenes to this locus, that we could get constitutive and persistent expression of our transgenes. And in fact, we showed that we could successfully differentiate ES cells into expressing luciferase from the GAPDH locus into stem cell-derived beta cells, as is characterized by NKX6.1 and C-peptide expression. And that when we transplant these cells into either immune-deficient mice, which are DKO mice, double knockout mice, or immune-competent mice, we can see their persistent expression in the immune-deficient mice, whereas in the immune-competent mice, they're completely wiped out within about 10 days. And that is a xeno-rejection that you see in the ICR mice. So that's human to mouse, xeno-rejection. And so we were quite confident that if we used a similar strategy, we could introduce tolerogenic molecules also at the GAPDH locus to engineer hyperimmunogenic stem cells. And so we actually generated a very similar construct where we introduced luciferase as a reporter of cell viability, because ultimately we want to determine whether our cells are surviving or dying, both in vitro and in vivo. And luciferase is a great reporter of cell viability, and also co-introduced tolerogenic molecules. And in this case, I'm going to use HLA-E, the HLA-E long chain fusion as an example. And then what I did was I then systematically knocked out beta-2M in these cells such that they were deficient for all class 1 molecules except for the long chain fusion. So these cells now either express, so we've generated 3 cell types now, wild type cells that express luciferase and that still express all their MHC molecules. Beta-2M deficient cells, so which are a control line that express no HLA. And then these Beck cells, which express luciferase and only the HLA-E single chain fusion, but no other class 1 molecules.
Dario, why did you guys go for HLA-E first? I mean, did you ever think about HLA-G, right? Because that's a protective feature at the placental trophoblast from kind of create, it's helping create that immune privileged space of the placenta.
Yeah, that's exactly right. So we did test a number of HLA molecules. We also tested HLA-G, and we found that HLA-E had a stronger effect versus HLA-G. So that's why we kind of went with HLA-E over HLA-G. But you could also introduce HLA-G as well, because at the end of the day, I think ultimately it's going to require multiple transgenes to really achieve true hyperimmunogenicity. But in this case, we've gone with one to kind of show proof of concept.
Okay, cool.
Yeah. And so then ultimately what we wanted to do was show that we could differentiate all of these gene modified lines into SC beta cells using our differentiation protocol. And so this data here just shows that we can differentiate both the wild type, the HLA deficient, and the Beck line into stem cell derived beta cells, and that there's no significant difference in the number of beta cells that we generate from all of these 3 lines. And that's really important to kind of show that. We also then characterize these cells in terms of their HLA expression and show that only that wild type cells upregulate HLA expression upon interferon gamma stimulation, whereas the other 2 cell types, the HLA knockout and the HLA-E expressing cells, do not upregulate HLA class 1 expression. And that HLA-E expression is constitutively expressed only on the Beck cells, Beck beta cells, whereas in the wild type cells, it's upregulated, whereas in the basal condition, it's quite lowly expressed. And then finally, in the bottom panel here, I wanted to also confirm that these cells express luciferase at essentially equivalent levels. And so we just did a comparison of luciferase expression in all these 3 lines. And as you can see, there's overlapping luciferase expression in SC beta cells derived from all of these 3 lines. Let me move forward. So next, ultimately, what we wanted to do was test these SC beta cells in vitro co-culture assays first. So what we did was we isolated various immune cell subsets from peripheral blood from various donors, allo donors, not necessarily diabetic. In fact, we're blinded to whether they're diabetic or not. So for
human donors.
Yeah, they're just human donors, allo human donors. And then we co-cultured them with the SC beta cells derived from these gene modified lines. And they used luminescence as a readout for cell survival. So if the cells still light up, they're alive. And if there's no light, they're dead, essentially.
Yeah, that's an elegant, but also very short and sweet sort of way of doing things.
It is. It's very nice in the fact that you can also translate the same assay in vivo. So you're not working with 2 different assay systems. You're using the same assay both in vitro and in vivo.
Nice.
So essentially what we did was we co-cultured them as described, and then determined percent survival based on luciferase expression and relative to the addition of no PBMCs. So you would expect that in the absence of any PBMCs, you'd see no killing of your SC beta cells. And what you can see in this figure here, that at multiple effector target ratios, in blue is cells that were co-cultured, or SC beta cells that were co-cultured with naive PBMCs, and in red are activated PBMCs. And you can see that in the naive condition, that beta-2M knockout and Beck expressing beta cells are somewhat protected from PBMC destruction. And you can see this at both effector target ratios, whereas activation of PBMCs essentially results in a wipeout of all the SC beta cells derived from the gene lines. And we can also see a similar profile, but it was not as strong, with CD8 positive T cells. So when we isolate CD8 positive T cells from the same donors, repeat the assay, we can see a similar type of profile and protection against CD8 positive T cells. The next thing we kind of wanted to look at was their protection against NK cell cytotoxicity. And the reason being was that once you eliminate HLA expression, the dogma suggests that these cells would be then susceptible to NK cell cytotoxicity, which was also one of the reasons why we introduced HLA-E into the cells to kind of be an inhibitory signal to NK cells in the absence of any other classical HLA expression. But what you can see in this, this is some data from a recent paper published in Stem Cell Reports of wild type and HLA deficient platelets. And what they did was that they looked at NK cell activation when co-cultured with primary human NK cells. And what they found, there was no significant difference in NK cell activation between wild type and HLA knockout cells, whereas K562 cells, which are known to be stimulatory to NK cells, did activate NK cells. And in a similar study also published recently from Sonja Schrepfer's lab, they showed that human primary NK cells could not destroy HLA deficient endothelial cells. And so if you take a closer look at this top panel here, you can see wild type cells, HLA deficient cells, and their transgenic line, which was a HLA CT2A CD47 knock-in line. And as you can see, that there's no significant difference in the survival of these cells, but only when you add in interleukin-2 to the primary NK cells do they become activated and then start to kill HLA deficient cells. And in fact, we saw something similar with our SC beta cells. So we tried to repeat these assays. We co-cultured our SC beta cells with two different NK cell systems. The first system was using the NK92MI cell line, which is an IL-2 independent cell line. It's been retrovirally transduced to produce its own IL-2. And what you can see is that the HLA deficient cells, or the beta-2M negative cells in this system, are somewhat more susceptible to NK cell cytotoxicity, whereas the HLA-E knock-in cells are protected from NK cells. And then we have K562 and Raji cells in there as our positive and negative controls, respectively. However, when we co-culture our SC beta cells with primary NK cells that have also been pre-treated for 5 days with IL-2, which you would anticipate would destroy HLA deficient SC beta cells, we actually see no significant difference in NK cell cytotoxicity against all three gene modified lines, which is surprising when you think back to Sonja Schrepfer's work, where they showed that if you stimulated with IL-2, you could induce cytotoxicity against HLA deficient endothelial cells. So this was somewhat surprising to us, and we wanted to investigate this a little further. So we went back to some of these single-cell RNA-seq data sets that Adrian in our lab had developed. And we wanted to look specifically at the activating and inhibitory ligands in the SC islet cells that we've generated and compare those to primary human islets. The reason being was that NK cell cytotoxicity is actually dictated by a balance of both inhibitory and activating signals. And so in the absence of potentially any activating signals, there's no reason for an NK cell to become activated and destroy. And so you can clearly see here when we compare activating expression of activating ligands versus inhibitory ligands, that SC beta cells or SC islet cells and primary human islet cells display a quite strong and distinctive inhibitory phenotype versus activating phenotype. And this may explain why our SC beta cells are somewhat intrinsically resistant to NK cell cytotoxicity within the context of HLA deficiency. We also then wanted to confirm the RNA-seq analysis by looking at protein expression by FACS. And so we confirmed the expression of some of these molecules on the surface of our SC beta cells in all three lines. And you can see that our cells express CD47 and nectin, PVR as inhibitory ligands, but that they don't express many activating ligands. And then finally, what we wanted to do, I think, which is really kind of the proof of the pudding, was transplant these cells into humanized mice. So these are HLA deficient mice, so they're double knockout mice, and they've been humanized with HLA-A2 mismatched PBMCs. They've also been transplanted with either the wild type or the HLA-E expressing SC beta cells. And as you can see, after 35 days, our HLA-E expressing SC beta cells show significantly improved survival versus the wild type cells. And so this was really exciting to us. And we're going to be following up on some of this data with trying to replicate these experiments in a diabetes context as well. So in a hyperglycemic context.
That's really interesting. Oh.
Yeah, so I think just to conclude, I think what we've shown now is that if we target transgene expression to the loci of housekeeping genes, we can solve the issue regarding transgene persistence. We can successfully now successfully differentiate gene-modified human embryonic stem cell lines into SC beta cells with similar efficiencies to the unmodified cells. We've also shown now that HLA deficient SC beta cells are resistant to T cell and NK cell cytotoxicity in vitro. And that surprisingly, SC beta cells intrinsically possess an NK cell inhibitory ligand profile, and that the lack of these activating ligands could spare them from the missing self cytotoxicity. We've also shown that HLA deficient SC beta cells now possess an in vivo survival advantage over wild type SC beta cells. And ultimately, I think this is one of the most important kind of conclusions, that we need to start to consider the intrinsic ligand profile of the differentiated cell product that we want to be producing when determining the set of genetic modifications that may be required to engineer hyperimmunogenicity. So not all cell types are going to be as essentially immunogenic as others. And so if we consider SC beta cells versus potentially endothelial cells, which are more immunogenic, the set of modifications may be somewhat reduced to engineer hyperimmunogenicity in SC beta cells versus a cell type that's more immunogenic. So I think that's kind of really important to consider moving forward as well. And finally, I just want to thank, obviously, all my colleagues in the Melton Lab and those who actively participated in this research as well. And so I'm happy to take any questions.
Fantastic. Well, yeah, we definitely would like to take questions from the audience. You guys can unmute yourself as an audience member, or you can put your question in the chat, whatever you prefer. I had a couple of questions. I wondered, can you hypothesize about your findings that these SCB inherently lack these activating ligands? What is it that makes them behave this way?
Yeah, so we're unsure at the moment as to why they're somewhat hyperimmunogenic. SC beta cells have been shown to be hyperimmunogenic in the past. I think there's a paper, I can't remember, Zaldumbide et al., that showed that SC beta cells are somewhat hyperimmunogenic. But what was really interesting was that the primary human islet data seemed to match that as well. So primary human islets seem to be somewhat hyperimmunogenic as well. At the moment, we could only speculate as to why this may be the case, but it seems to serve us well in terms of engineering strategies.
Yeah, no, that's it. I mean, it'll be really interesting to find out sort of, more about that as things progress. I wondered, have you guys thought about or looked at any of these HLA deficient SC beta cells in context of viral infection like CVB? What do they do? How do they behave in that context?
Yeah, so that's really important when we start to think about safety. We haven't looked at that yet, but I think that's going to definitely be an avenue of testing downstream, because safety is going to be the most important thing at the end of the day. Right. And I think we need to hammer that home to anyone that's working on generating hyperimmunogenic lines, is that ultimately safety has to be one of our readouts as well. And I think, like you mentioned, testing these cells against viral evasion, potentially even forcing them into forming teratomas to see what they do once they form a teratoma. Yeah. We don't want them to form teratomas, but we need to consider that as being a potential. Well,
if they did, and you had some kind of alert or some kind of way to detect that and could remove the
Yeah,
insert, well, then that's still could be okay, right? If
exactly.
things started to go in a different direction than expected or wanted, then you could have some kind of litmus test embedded in the implant and be able to remove it.
Exactly.
Still, even if you could do the implant for, a couple of months and then get another implant, still would be a lot more, it would just be so beneficial to have that level of control versus what many type 1 diabetics have currently.
I agree. Yeah.
And in the audience, would anyone like to unmute themselves? I really would love to get some conversation flowing. Okay, nobody wants to. They're there, but they don't want to.
That's okay.
That's fine. And then we have had Adrian Veras join us before, and Nayara Leti, which I think, I don't know if she was on the specific project, but I know the lab is, really quite connected to the Vertex group. It is interesting that ViaCyte now is connecting with the CRISPR group. And
Yeah.
I mean, do you see some kind of symbiosis with Vertex and maybe an in-house sort of CRISPR situation or external? I mean, is that a direction they're going? You have any idea? I mean, you might not even want to say or might not know, but I just thought I'd ask
look,
it.
that's something I don't know yet. I think that's probably on Doug's level at the moment. But it definitely is interesting to see these companies collaborate with these genome editing kind of focused groups. So we know, as you mentioned, ViaCyte's been in cahoots with CRISPR Therapeutics, and I think Vertex have recently mentioned that they're joining forces with Beam, and so I'm sure that that's probably down the road for them as well.
Yeah, I think it's all good news. I think collaboration, I mean, with some healthy competition right between the two camps could really drive, the research in the area forward.
Yeah.
So it's great. It's really great.
Yeah.
And what's next for you? You're going to publish these results. I hope that's on the way so everyone can read all the finer details of the paper.
Yes.
That'd be cool.
Yeah, so this is all in preparation, trying to wrap this up now with the last models, as I mentioned, putting these cells into diabetic humanized mice. As you can appreciate, these are challenging models to develop.
Yes.
And I think the next step would be testing these in a more clinically relevant model. So going maybe into non-human primates and trying to really test true allo rejection, because the models that we have, or the mouse models that we have available to us right now, really bias certain components of the immune system. So we're not truly able to test all our engineering strategies against a complete human immune system or a complete allogenic immune system. And so I think that's going to really be the proof in the pudding for all of these engineering strategies and hyperimmunogenic cells, is working with an in vivo model that truly recapitulates allogenic rejection. Yeah,
it definitely seems right. I mean, there's this whole sort of like islet on a chip and organ on a chip approach, but it might not fully capture the environment, shall we say. So I think, that said, I mean, Dario, this is really interesting work, a very thorough and very, very detailed work that you've done. And I really appreciate you sharing it with us. It's fantastic. We really can't wait to see what you do next.
No, thanks for having me, and happy to chat to anyone after.
Yeah, for sure. We'll share your email should anyone like to reach out, and we typically have many views on our both our YouTube channel, and we share this as a podcast as well. So I'm sure the word will get out and people will enjoy what you shared with us today at other times. So thanks again. Have a great rest of your day.